Hello. It is an enormous pleasure to be here for another Coffee and Science. And this is a special one, I'd say, because this time the person who is always with me presenting the speaker is the speaker himself. And just before I introduce Darlan as the speaker for today, I'd like to say for those who are watching us for the first time, what the Coffee and Science is very briefly. We are a space at the University of Oxford where anyone doing research on Brazil can present some of their ideas. So we usually have PhD students and professors at the university and researchers who have an opportunity to gain feedback and build up collaborations presenting work on Brazil. We also eventually have visitors and collaborators who are outside Oxford, but who also have research focusing on Brazil and are in collaboration, partnership with somebody here at the university. And today we have Darlan Candido, who has graduated as a pharmacist from the Federal University of Ceará, and he holds a master's in allergy and immunopathology from the University of São Paulo. Darlan is currently a PhD candidate in zoology at the University of Oxford, and he applies genome sequencing to understand virus evolution and spread during outbreaks of public health concern. He's currently working on SARS-CoV-2 outbreak in Brazil, and he's going to be talking about it. My name is Andresa. I'm the director of the Brazilian Studies program, and I will be sharing the talk with you today.
Yeah, thank you very much, Andresa. It's a pleasure to finally be here speaking about my own research and about research of other people too on the coffee and science. It's been so far such a great pleasure to be sharing it with you, but now actually presenting something, it's going to be quite different and a new experience for both of us. So I'm going to share my screen and remove you. And let's share the screen. Okay, so today I'm going to be speaking about the genetic diversity of SARS-CoV-2 in Brazil. And as Andresa said, I am a PhD candidate in zoology at the University of Oxford. And since last year, both Andresa and I have been working on the Brazilian response to the COVID-19 pandemic and the epidemic in Brazil. So I'm going to be sharing some of our results so far. I'm going to be giving you an overview of the epidemiology in Brazil and also obviously the genetic diversity of the virus in Brazil. And at the end, we're going to be speaking a little bit about vaccines and what all of this means for the vaccination programs that we have in Brazil too. I might actually leave you with a few more questions and answers, but I guess that's science, right? So let's start. Okay, so first I'll give you an epidemiological context of what was the beginning of the SARS-CoV-2 epidemic in Brazil and the whole process of notification and the difficulties in notification systems and reporting systems in Brazil. So since years ago, Brazil has had the CVAP-GRIP, and the CVAP-GRIP is a system for notification of flu cases. It's a flu surveillance system in Brazil. But for the SARS-CoV-2 epidemic, and anticipating that this would be a problem in Brazil, the Ministry of Health created the REDCap, which is a notification system that was a reporting system specifically designed for SARS-CoV-2. However, there were some problems with the system, and it was discontinued in late March, as you can see. And then there's an added complexity to it because the reporting of SARS-CoV-2 cases in Brazil was split between two different systems according to severity of the cases and according to where the cases were being notified from. Why am I talking about this? It's just to give you an understanding of the complexity of the response in Brazil, starting from the reporting systems. So by the end of April, we already had more than 60,000 cases in Brazil and around 8,000 deaths. And this is the picture that we're going to be talking about when I'm telling you about the first 2 months of spread in Brazil, which is quite different from the current picture we have in the country, which we will be discussing a bit later. So in this first few days of spread in Brazil, until actually the first month, until the end of March, what we could see is that in accordance to our predictions, we actually saw a great majority of the importation cases in Brazil coming first from Italy, and then we saw a lot of cases coming from the US later on, after, of course, the epidemic was picking up in the US too. And this is interesting because we predicted that this would happen based on the pattern of flights, I think international flights to Brazil, and also the number of cases in the prevalence in these different countries. We also saw early on that there was an underreporting of confirmed cases. So the blue lines here that you can see below, they are the number of cases. So the full line, and the dotted line is the number of SARI cases. SARI is severe acute respiratory infection, which is the main manifestation of the COVID-19 disease. And what we can see here is that for the flu epidemics in Brazil, these numbers were much smaller than what we saw last year. And for the full line here, we see COVID-19 cases, but SARI cases without any etiological agent, so SARI cases to which the cause we don't actually know, were much higher than the number of COVID-19 cases. So we have a great understanding of actually the number of cases being reported, at least at first, was much smaller than the real number of cases of COVID-19 in Brazil. And finally, we also noticed that the epidemic was spreading from cities with a larger population first, and then going to cities with smaller populations in Brazil, which is like the process of spreading from mostly capitals and big urban centers to the countryside of each state. And there was actually one of our papers looking at the lack of coordination when it comes to the response and the implementation and easing of non-pharmaceutical interventions, such as lockdowns and use of masks and social distancing, closure of commerce, non-essential commerce, and schools, and so forth and so on. And in this paper, we particularly worked with Andres. Andres is actually the first author of this paper, so I should actually ask her to explain it. But it's going to be very quick. So in this paper, which is really interesting, we looked at data from more than 4,000 municipalities in Brazil, which is around 70% of all municipalities in the country. And we had information on implementation of non-pharmaceutical interventions, the dates in which they were implemented, and also the dates in which they started to be eased for each of the municipalities that responded. So in here, we're basically seeing a focus on the state of Minas Gerais, which is the second largest state in the country in terms of population, the first in number of municipalities, and also the states in which we had more respondents. So what we can see here is that municipalities are colored according to the month in which they eased non-pharmaceutical interventions. And what we can see is that there is a mixture of different months here. But the importance of this is that even in municipalities which share borders, some of them were easing NPIs in March, while some of them were easing them in June. But obviously, the easing NPIs in one municipality has an effect in other municipalities when they share borders. So there's like this disconnection in the response in Brazil. What we also saw, and this was in our science paper, we were looking at the reproduction number. And just for people who might not be familiar with it, the reproduction number is basically the average number of people that a person who is infected will pass the virus on to. So if the reproduction number is 3, it means that if I'm infected, I will transmit the virus to 3 more people on average. So for this study specifically, we looked at the city level. So we looked at Sao Paulo and also looked at Rio de Janeiro. And for both cities, what we saw is that before the implementation of non-pharmaceutical interventions, the reproduction number was between 3 and 4, meaning that an infected person would infect 3 or 4 more people on average. However, after the implementation of NPIs, what we saw in Brazil was a huge decrease on the transmission, on the reproduction number. In Sao Paulo, we even had the reproduction number going lower 1, which is the ideal. When the reproduction number is below 1, it means that the epidemic will start decreasing, and hopefully it will cease. However, what we saw in both municipalities is that it reached a steady state of around 1.5. And that's why we saw the outcomes of this epidemic in Brazil, which is a large and very long steady state in Brazil with a high number of deaths, which started to decrease at around September and October, but it's now picking up again. With the increase of the reproduction number. The current situation was quite different from the one I described at the beginning of the presentation, because now we have almost 10 million cases in Brazil and more than 230,000 deaths. So it's painful to watch. But what we also noticed is that in some cities in Brazil, such as Manaus in the Amazon region, we saw high attack rates of the virus, meaning that for Manaus, it has been estimated by our group that up to 3/4 of the population, 75%, might have already got infected. And if you have been watching the news or reading the news about Brazil, you'll see that in Manaus, they're having a really severe second wave, which is even worse than the first one. And this was highly unexpected because of the fact that we estimated that up to 75% of the population had already got infected. But we can discuss this a bit more later when we're talking about lineages. So this was the epidemiological and actually public health response overview that I had for you. And this is interesting so everyone can be on the same page in terms of what happened in Brazil at first. But now I want to make a link with genome sequencing in our group and how we started doing genome sequencing, helping with the response in Brazil, but also what we can conclude from it. So for those of you who are from Brazil, you might have seen in the news at around March and April, talking about Esther and Jackie, and all of the group from Esther 2 and Ingra and Flavia, because together, we sequenced the first 2 genomes of Latin America from Sao Paulo in less than 48 hours. And this was a huge thing for Brazilian science. And we also released a report showing that these cases were linked to most likely to the epidemic in Italy. And these were the first 2 reports of the virus in Brazil. That's how we started playing a role in the pandemic in the country. And later on, we decided to go ahead, and Andresa will remember this. We had meetings every day with around like 40 people from all of different areas, political science, virologists, epidemiologists, mathematicians, and so on, talking about how we could understand what was going on in Brazil and how to produce important information for the country and for the response. It came to its peak when we released our science paper on the evolution epidemic spread of SARS-CoV-2 in Brazil. And here's just so you have an idea of the number of people that were actually working for this paper to become true. What's really interesting about it is that by putting together a task force of people for genome sequencing in Brazil, and here we have Esther's group at the University of Sao Paulo, we have people at Unicamp, we have people at Federal University of Rio de Janeiro, of Minas Gerais, and also from the National Laboratory of Scientific Computation. And putting all of this task force together, we were able to sequence 427 samples from 85 municipalities from 18 states in Brazil, so we could have a good idea of the local transmission of the introductions and local transmission in the country. So in here you will see the representativity of our sampling from Brazil according to time and also according to the different Brazilian states. And our final data set had 490 genomes because we also used 63 genomes that were available on the SARS-CoV-2 platform, GISAID. And in here, we also acknowledge the other authors for that. So using this data set of 490 sequences, it was very interesting to see that we were able to estimate more than 100 SARS-CoV-2 introductions to Brazil, because this is almost like 20% of all of the sequences in the country. What were telling us a story of importation, and then it tells us how important international importation is for the actual pickup and spread of the epidemic in Brazil. So this figure here might seem very complicated. It's a phylogenetic tree. So basically sequences in here, they are clustering according to the mutations they have, in a very simple way to explain it. And the tree itself is colored according to where the sequences come from. So anything blue means that the sequence was collected in a country different from Brazil, other than Brazil. And anything in red means that these sequences are coming from Brazil. And when we see like really large red clusters, these are the clusters of local transmission in Brazil, and we have 3 that we identified in this paper. So we have clade 1, clade 2, and clade 3, and I'm going to tell you a little bit about them. What was also interesting to see was that the introduction of the virus and the local spread of the virus in the country was estimated to be between mid-February and late February. And the first cases in Brazil were detected on the 25th of February. So the surveillance in Brazil by that time was relatively quick on picking up the introduction of the virus in the country and also the local spread. And again, this is the information that we had by 30th April 2020, and we'll talk about what's happening now later on. So what actually those 3 clades mean for the transmission in the country? So clade 1 was telling us the history of transmission in the state of São Paulo. So the pink color here means São Paulo. So mostly the state of São Paulo, and this lineage is the one called B1128. When it comes to the clade 2 actually represented most of the local transmission across the country. There are lots of sequences from São Paulo, from Rio, and from Minas Gerais, but sequences from other states too. So, and this is the lineage B1133. And you'll understand why I'm telling you which lineages these are. And then the third clade was the smallest one back then. It's the clade from Ceará state. And Ceará had a completely different diversity than the one that was circulating in the rest of the country. So this was really interesting to see. And this is probably linked to the international connections with other countries in Europe that were different from the ones seen in Southeast and São Paulo. So we also concluded that there were 2 initial phases of the spread of the virus in the country. So the first one was a phase that was marked by the intra-state transmission. So transmission was happening basically between cities in the same state, shorter transmission. While the second phase, which is markedly after the 21st of March, was happening in a different way, in which we had an increase in the interstate and inter-regional spread. So we saw more transmission between states and between regions. And you can see that in this figure by these long lines that go all across the country, mostly from Southeast to other regions and other states in the country. But why is that? Why do we see such a big difference from one date to another one? And what was interesting for us to see when we looked at air travel data, so human mobility, is that all of the flights had an impact, were impacted, by the decrease in the number of passengers. However, flights with longer journeys were less affected than flights with shorter journeys. And this is a bit logical to explain in the sense that it's much easier to cancel a short flight and get on a car and travel across the state than having to take a car journey across Brazil from São Paulo to Ceará or from São Paulo to Manaus. And because of that, we saw that after the implementation of non-pharmaceutical interventions, the average journey for a passenger, for an air travel passenger in Brazil, actually increased, meaning that people in general were flying less, but the people that were flying longer distances, such as Ceará or Manaus, and so forth and so on. Well, but then this was the main conclusions that we could take from the genomic data that we sequenced back then. But right now, especially in the media and the scientists and reports and everything, we're talking a lot about mutations and lineages, right? And I believe that for most of you, sometimes it's difficult to understand, are all mutations and lineages worrisome? What do they actually mean? And the easy answer for this is no, they are not worrisome. Mutations are something common that happen in every living being when they are replicating their genetic materials, basically making copy of their DNA or RNA and so on. The thing about the coronavirus is that the SARS-CoV-2 is that it is an RNA virus. And RNA viruses, they evolve, they acquire mutations much more often and much faster than our cells do. And that's why we keep tracking the evolution of them, because these changes, they have impact on the transmissibility of the virus, and the severity, on vaccines, and so forth and so on. So just so you have an idea, right now, globally, we have more than 900 lineages of SARS-CoV-2. Should I be worried about it? No. As I said, most mutations are something common, and lineages are going to come because there's a lot of people infected in the world, really. But most mutations, actually, they don't mean anything for the virus or for us. And some of them are actually bad for the virus. There's only a short number of them that are actually impactful and might be of concern for us. And to track these lineages, we developed, and by we, I mean scientists have developed a nomenclature for the different lineages according to the mutations that they have. And that's why we have this long number such as like B117 or P1, and that we will be talking about. It's because it's easier for us to understand, okay, so we're talking about this specific lineage with these mutations that is spreading across these countries. But what is the actual SARS-CoV-2 diversity in Brazil at the moment? So we now have almost 2,500 sequences. We have 2,500 sequences of SARS-CoV-2 from Brazil. So that's the information that we have to understand what's going on in our country. However, most of this information comes from sequences that were collected until July last year. So there's not as much information from what's happening right now. This has been changing the past month, and it's increasing. So we're getting a really good idea of what's happening now. But that's why the earlier lineages that I told you about, the one spreading in São Paulo and the one spreading in Brazil, are still the main lineages when we count the number of sequences that have been released since the start of the pandemic. However, we have 3 lineages in Brazil that are considered lineages of concern and that are increasing in their frequencies. So we're talking about P2, P1, and B117. And I know that these numbers and these names might mean nothing to you, but I will explain a little bit on the next slides. So B117 was first detected in the UK. Some people call it UK lineage. We try not to call it UK lineage or Manaus lineage or South Africa lineage because of the stigma that it brings. But it was first detected in the UK in September last year. And the first reports about this lineage, they actually came out close to Christmas last year. So it has now been reported in more than 75 countries. Why is it so important? And here again, we have a phylogenetic tree, and this long line here is a long branch that we call. And this long branch means that it has acquired many mutations, and many more actually than we would expect it to have acquired, given the normal rate in which SARS-CoV-2 acquires mutations, which is 2 to 3 mutations per month. And this is why it first caught our attention. The other issue is that it has acquired 8 mutations in the spike protein. And the spike protein is the protein that has the region of the virus that interacts with the receptors in our cells. And so it's really important for the entrance of the virus in our cells and so forth for transmission. So we know now that it's more than 50% more transmissible than previous lineages. There's evidence that it might be more lethal too, but we haven't come to a conclusion yet. And the key mutation that it has is the 501, and you'll see it come again in the presentation. But the 501 has been linked to a higher transmission of the virus, and so it's linked to this higher transmissibility seen here. So it has now been identified in at least 2 Brazilian states in the federal district, and our group described the first 2 Brazilian cases. Ingra is the main author of this paper. Maybe she's watching. Okay, so this was the lineage from the UK that was introduced in Brazil. But we have P1, which is the lineage that was first described in Manaus in Brazil. And it was actually first detected in Japanese travelers that were returning from Manaus in January this year. But then our group also has described the lineage from the sequences that we have from Manaus too. So now we have a better understanding of what the frequency of this lineage is in Manaus. So what's worrisome is that this new lineage has acquired 10 different mutations in the spike protein, so the protein that interacts with our cells. There is evidence for it to be more transmissible, but we haven't come to a full conclusion yet. We need more experiments for that. But if you check here, this table on the right-hand side, you'll see that from November to March, there was no P1 in Manaus. In December, P1 was already accounting for half of the infections in Manaus. And in January this year, it's accounting for at least 85% of all of the infections in Manaus. So the frequency of this lineage is growing very fast, and it's outpassing the other lineages that are already circulating in Manaus. And this is really worrisome and it's indicative of a higher transmissibility. It has 3 key mutations, which is the same 501 that we saw for the UK, 484 and 417. And I'm going to focus here on 484 because 484 has been linked to evasion of the immune system. So meaning that the previous infection or a vaccine might not be as effective. And we'll touch on that a bit later. But these 3 mutations are also present in the B1351, which is the lineage identified in South Africa. So you might have heard of it already. B1 has been identified in at least 4 Brazilian states, and there's at least 1 confirmed case of reinfection caused by this lineage. And then we also have P2. P2 hasn't called that much of attention. It has many less mutations. So 1 only mutation, the spike, but this mutation is in the same one that we see in P1 and in the South Africa lineage is 484. So as I told you, it has been linked to the evasion of the immune system. And it's now presenting quite a few Brazilian states, if not all of them already. And there's at least 2 confirmed cases of reinfection that have been linked to this lineage in the country. Okay, but I feel like the question that all of you might be thinking of is, so what is the importance of this for vaccines? Does this mean that the vaccines are not going to work? We're all going to die? So what's going to happen? And luckily, the first few, the first pieces of information have started to come and from clinical trials of vaccines. And now we know that, let's start with B117, which is the lineage identified in the UK. For B117, there is a decrease in the efficacy of the vaccines when compared to previous lineages. However, this decrease is not as marked, and it means that vaccination will probably work just the same as it would work for the previous lineages that were used actually to design the vaccines. However, when it comes to the lineage identified in South Africa, B1351, we see much more of a marked decrease on the efficacy of these vaccines. And this is really new, like fresh data. So for Novavax, we see that it decreased from 89% to 60%. For Johnson & Johnson's vaccine, it decreased from 72% to 57%. And for the Oxford AstraZeneca vaccine, even though they have a small sample size, the efficacy drops to 10%, which is essentially none. So that's why I think this week, I'm not sure if it was yesterday, South Africa has announced they won't be using the AstraZeneca vaccine anymore, and they will be using the Johnson & Johnson vaccine instead. So what does this mean for Brazil? We don't know, but the issue that we know so far is that the South Africa lineage and P1, which is the Manaus lineage, they share 3 mutations. So we're likely to see a decrease in efficacy for some of the vaccines for the P1 lineage, but we have no data on that yet. So there is a summary of these lineages and variants of concern and mutations and everything. So if anyone is interested, this was taken from Eric Topol's Twitter account. If you're interested in SARS-CoV-2, you should follow him because he's a great physicist and he's always bringing new information and making this kind of summaries, which is really nice. And that's what I had to present today. So kade is an ever-growing network of people who are interested in studying the spread of diseases using genomics and epidemiology. It was born as a center to study arboviruses, but because of the COVID-19 pandemic, we have been focusing on COVID, although we're still working on arboviruses too. So there's a lot of people who are actually not here, who I should be thanking to, but thank you to everyone that helps in all of this work, and thank you to the audience too. And I'll be taking questions now.
Thank you very much, Darlan, for this amazing presentation. And very insightful because you go from the very beginning of the pandemic Brazil, the early days, and build up until we get to the vaccine and the new variants that we have to deal with. So going back also to the early days of COVID-19 and coming to where we are now, when the outbreak started in Wuhan, in China, we remember some measures to isolate that city. It feels very radical. It felt very radical at the time. But in a country the size of China, perhaps we can think of the size of the outbreak had they not done some control measures. Now, you mentioned some of the NPIs, the non-pharmaceutical interventions, and cordão sanitário. I forgot how it's called in English. It is
sanitary barrier.
Something like that, where you check borders and you create certain controls. It was not one of the most popular NPIs. And again, you have the issue that not always roads is the main transportation. So you also need controls in airports, and there are different authorities who control each of these areas. So policy struggles on the side, and focusing on the pandemic, we have not seen any concern about Manaus and its potential spread. As far as I know, people can still come and go easily.
Yeah.
If you can comment a little bit what that means for control of the pandemic and what other countries are doing on that.
That basically means that the virus is probably freely spreading across the country, not just B1 lineage, but B2 and also B117, so the lineage identified in the UK, which was detected in São Paulo in late last year. So when we don't have any sort of checkpoints or sanitary barriers or anything like that, we're freely allowing people to move, and as a consequence, we're freely allowing the virus to move too. So it's very likely, for example, that B1 is already all over across Brazil, also considering that patients from Manaus had to be transferred to other hospitals in other states in Brazil. Their families are very likely to have followed them because obviously you want to be close to your loved ones. and that's how the virus travels and how it spreads and starts all the outbreaks in other states. For example, I'm from Ceará, so I'm very close to what's happening now, but we're starting to see an increase in the number of cases and deaths again there. And talking to epidemiologists and to medics, they are telling me, Darlan, this looks very different from what we saw in the first wave, so it's probably P1 that's already arrived in our state. So it's very likely that's already across Brazil, and it means that we might be hit by an even harder second wave than we were for a first one. The issue with that, of course, is will vaccines work? It's been linked to reinfection, so we'll have to see what happens. But again, we are watching what happens in the country when there's no measures in place to stop the spread of the virus. We saw this once happening last year, and we're going to see it now again with the new lineages, and it's very sad.
Yeah, you end up as answering my second question, which was, so why are we not seeing something similar to Manaus yet? But then you just mentioned that in Ceará, it's starting to show first signs, and it's indeed very concerning.
Yeah, and it's actually happening in other places too. So Pará is going through something very similar right now, and E1 has already been confirmed there. Roraima is also close to a break in the public health system there, and I think we're going to see more and more of this happening because we were already running low on ICU beds because of the regular lineages. So if P1 turns out to be more transmissible and also to be causing reinfection in basically people that have already been naturally infected the first time, then we'll see a huge increase in the number of cases.
Very concerning. So maybe get some from the chat.
Yes. So we have one question at the chat. And if anyone is interested in asking more questions, please do. And if something was not clear, please ask me. I know it can be a very dense topic. But Isabella Gomez is asking, Darlan, you mentioned that it's normal for the mutations to happen. My question would be, does this mean that we would need to be vaccinated every year to keep up with the variants? And Isa, that's a great question. And that's probably, and I'm saying probably, that's probably what will happen if we come to confirm that really all the lineages have been affecting the efficacy of the vaccine, as we have seen already for the South African lineage. So if the first results for P1 come out, and we see that, yes, the efficacy of the vaccines are just dropping really low, what was seen now for the Oxford AstraZeneca vaccine, then it might mean that we have to be vaccinated every year. What is the other concern here? So what should we be looking at and we're trying to do? If we stop, the more the virus spreads, the more likely we are to find mutations because we're giving more and more chances for the virus to mutate every time it infects other people. That's not the only one cause, but so if you can imagine that the virus will mutate every time it replicates, so the more people it infects, the more there's virus replicating, and the more prone we are to finding new mutations and for them to spread. So if we decrease the number of people getting infected and the incidence of the virus, if these vaccines start to work and some people are already protected, then we might decrease the frequency in which we see mutations. So what I'm saying is that it might not be every year that we need to be vaccinated, but something like this wouldn't be the kind of scenario which we would say, well, we don't expect that to happen. We see that very often. We see that already with influenza, so the flu, seasonal flu, and every year we have to take a different, to get vaccinated with a different vaccine for flu because of the different lineages that are circulating every year. The difference between the flu and SARS-CoV-2 is that the flu acquires mutation twice as fast as SARS-CoV-2 does. So that's why I'm also saying it might not be every year that we need to be vaccinated. But this is something that we will only actually know later on when we start to see the effects of the massive vaccination programs that we are seeing worldwide. I guess, Andressa, we don't have any more questions. So
then can I have another one?
Sure, definitely.
Okay, so regarding reinfection and P1, I know
Yeah.
there is one case described already in the literature
Yes.
of a reinfection, and I had a look at it, and there is quite a gap. I think the first infection was in March, and the
Yeah.
second one was in December. So, correct me if I'm wrong, but that case alone, it would still be a bit borderline to say immunity escape or just waning off the
Antibody response.
yeah, sorry.
Waning of the antibody response, the antibody level.
Exactly. And but combined maybe that data with the whole aspect of the pandemic, that's where you get this idea of reinfection. So can you explain a little bit more the cues that you follow to understand reinfection?
Yeah, so with P1, there's only one case that's been confirmed. However, with P2, which also has the same mutation that we're talking about for P1, we have two cases. And for the South African lineage, I don't know how many, but there's a few more that have been confirmed of reinfection. So, and from in vitro, so not looking really at people, but from in vitro studies in the lab, we have seen that some of the mutations in P1, P2, and B1351, the South African one, that they share, and they decrease the efficacy of antibodies to neutralize the virus. So meaning that if someone have already been infected, they have a higher chance to be reinfected by these lineages than with this mutation than they would have with a lineage that doesn't have this mutation. So there's a few pieces of evidence coming from testing laboratory, from what we observe in the general population, and also from what we're starting to see with the vaccine trials that were tested against the South African lineage, and we see very low protection, especially for the Oxford AstraZeneca. So it's a kind of a constellation of pieces that put together makes us think that. However, for example, for the situation right now in Manaus, in which I told you that it was estimated that 3/4 of the population have already been infected, there are 4 potential explanations for that. So the first one is that the estimates are wrong, and they were just overestimated, right? So it was not 75%, it was less. But even if it was less, the lower bound for these estimates is around 50%. So we should still not be seeing such a huge number of cases. So the second one would be that this lineage is more transmissible. And then if this lineage is more transmissible, which it might actually be, the herd immunity threshold would have to be higher, which might be the case. And then there's 2 other hypotheses which are bound to the evolution of the virus. So one of them is related to the evasion of the immune system, and the other one would be not related to the virus, but would be the waning of the antibodies. So all of them could actually be playing a role together. But as you said, at this point, with this one single case, we can't really tell if it was waning of the antibodies because we don't have that data for that patient, and or if it was the mutation or the lineage that's just evading the immune system. But then putting together these other pieces of information and of evidence, then we come to the conclusion that it might be evasion of the immune system. But we would need more data to actually understand that.
Well, very good, Darlan. And this also links to another point I have, but you may be seeing some questions on YouTube. But anyways, mine is no? Okay.
No more questions.
I'm like an infinite like source of questions. I can go on forever.
Yeah.
So that brings me to another point, which is genetic surveillance, right? So like we need more data to really understand what is happening. with P1 and other variants of concern. So please tell us, like, what is the proportion of the confirmed case that we can actually sequence? And I think in comparison with the UK, that I suppose is our standard
Yeah.
level now, like gold standard, like how far are we?
Yeah, so the UK is actually sequencing around 5 to 10% of the cases,
Wow.
and that's really, really high considering that
Mhm.
the number of cases that the UK has. But the UK is really off the curve in that sense. So it's the top country to compare to, but it's an unfair comparison to make at this point. However, for Brazil, let's compare Brazil with South Africa. South Africa has much less cases than Brazil, and South Africa has been sequencing more than Brazil has. Um, so for Brazil, we have almost 10 million cases, and we only have 2,500 sequences. So this is a really low number. It's not. I mean, there's other countries which have been sequencing less, but we need to step up and sequence more, and that's what we're trying to do right now. Obviously, there are barriers because Brazil is a developing country. Our reagents come from outside, and Brazil, all of the countries are fighting for this reagent, so there's lags in reagents to come. Sometimes it's difficult to work with the samples and to get samples to come from different parts of the country and so forth and so on. So there are limitations. All of them can be worked on. And we need really more genetic surveillance. And this is coming now, especially because of the situation in Manaus and because the virus is expected to have spread it. The new lineage is expected to have spread already to other states in Brazil. Then we're starting also to sequence from other parts of the country too. So for a while, we didn't have new information, and this was really worrisome. But for now, I think that we're starting to get a much better idea of the genetic diversity and the impact that it might have for the country. We need also other studies about coronavac and how it will actually
Mhm.
be working against the new variants in Brazil, both P2 and P1. But yeah, these are still chapters that we're going to start seeing very soon.
Thank you so much, Dalan, for all the complex explanations. So I don't know if there is anything else you'd like to say.
Just thank you very much for the opportunity. It's really nice to be here as a speaker and not just, as a chair with you. And I think, the Brazilian Society is doing an amazing job, like all of us together. And yeah, it's really nice to be here.
Yes, I think I take this opportunity to say thank you to you as the president of the Brazilian Society, but also the entire team of the Brazilian
Definitely.
Society. I don't think we would be able to have had any of these talks, never mind all of those we had, without the support of the Brazilian Society, which have been working closely with the Brazilian Studies Program, and we are really showing what this team of Brazilians and Brazilianists at Oxford are doing, and I think it's great to spread the word to the world, and really see like the comments from the audience and engage with people in Brazil and elsewhere. So thank you, Darlan, for being the chair in the meeting and the presenter today.
Yeah, thank you, Andresa. It was a great pleasure. And I think that's it, right? So goodbye,
Yes,
everyone. Thank you for watching. And if you have any more questions, I'm happy to help privately.
yes, we see you in 2 weeks' time.